Welcome back to this next video in which we are discussing the parathyroid glands and vitamin D. Okay, so in the previous video what we discussed is the two different forms of vitamin D, namely vitamin D2, also known as ergocalciferol, the structure of which is shown here, and vitamin D3, also known as cholecalciferol, the structure of which is shown here. Okay, now although these two molecules have different structures, as far as the body is concerned, as far as the actual biological activity in your body is concerned, they are utterly equivalent. They do the same thing. So it does not matter if you, for instance, don't have any vitamin D2 molecules, as long as you have lots of vitamin D3 molecules, those will cover the function of the vitamin D2 molecules. So they don't have separate functions, they do the same thing, and that's why we often just refer to them both under the umbrella term of vitamin D. So when we talk about vitamin D, we could be talking about molecules that look like this, vitamin D2, or molecules that look like this, vitamin D3. They're both going to do the same biological activity. Okay, right, so what I now want to talk about is what is this biological activity? Well, the first thing to say is that vitamin D is a fat-soluble vitamin, which means that it's not terribly soluble in water. Instead, it's much better dissolved in fat molecules. And of course, the reason for that is obvious when you look at the chemical structures of these two molecules. I mean, the only glimpse of polarity that either of these molecules has is the alcohol group of carbon-3. Everything else is incredibly non-polar and is therefore not going to dissolve well in water. So in order to transport these molecules around the bloodstream then, we are going to have some protein potentially to bind to them and transport them around the body, and indeed there is such a plasma protein that is there for vitamin D molecules to bind to so that they can be transported around the body, and this is known as the vitamin D binding protein. So in order for the vitamin D molecules and the derivatives of vitamin D molecules, which we're going to talk about, to be transported around the blood, there are vitamin D binding proteins in the blood which will bind vitamin D molecules, and that's the way in which they can be transported around the blood. Okay, so vitamin D binding proteins. Another little thing to be aware of with vitamin D being a fat-soluble vitamin is that it's going to be stored in the adipose tissue, the adipocytes of the adipose tissue. So adipose tissue, of course, we have lots of adipocytes where they are storing a huge number of fat molecules, generally triglycerides, in their lipid droplets. They can also store vitamin D in those lipid droplets. So you will have a large store of vitamin D molecules in your adipose tissue, and whenever vitamin D level goes down in the bloodstream, the adipose tissue will release vitamin D into the bloodstream. So when you eat a meal and get some vitamin D, or if the sun's shining and you're making vitamin D, that's when the adipose tissue will be taking up the vitamin D and storing it in their fat droplets. Whereas if your vitamin D level is too low in your blood, then the adipose tissue will be releasing the vitamin D into the bloodstream to keep your vitamin D levels at a certain level. Okay, right, so it's very difficult actually to become vitamin D deficient because of the fact that you have such a large store of vitamin D in the adipose tissue, and it will take you quite a long time to get through all of that and actually become vitamin D deficient, but it's very bad if you do become vitamin D deficient. Okay, so now let's actually talk about the function of vitamin D. So we have vitamin D in the bloodstream. What is it actually doing? Well, the answer is not that much, because vitamin D is not the active form. In order to actually do something, it needs to be converted into the active form, and the active form is 1,25-dihydroxyvitamin D. Now, in order to convert vitamin D into 1,25-dihydroxyvitamin D, we firstly need to convert it into 25-hydroxyvitamin D, and this is occurring all the time. So let me firstly talk about the 25-hydroxylation of vitamin D. Okay, so firstly 25-hydroxylation and then 1-hydroxylation. Okay, so the 25-hydroxylation of vitamin D involves putting an alcohol group onto a vitamin D molecule on the carbon number 25. And when I talk about a vitamin D molecule, I now mean either vitamin D2 or vitamin D3. So you can do this to both vitamin D2 and vitamin D3, and as far as the activation is concerned, it will have the same effect on those molecules. So we're now just going to talk about vitamin D rather than vitamin D2 and vitamin D3. So, 25-hydroxylation then majorly occurs in the liver. So let's have a hepatocyte here. Okay, so what's going to happen is the hepatocyte is going to take up the vitamin D molecule. And for short, I'll just abbreviate the vitamin D down to VitD here. So the hepatocyte here is going to take our vitamin D up, and then it's going to enzymatically catalyze the addition of an alcohol group onto carbon number 25. Now, what is carbon number 25? Carbon number 25 is this one in the side chain. So whether we're looking at cholecalciferol or ergocalciferol, D3 or D2, both of them have a carbon 25 in this side chain of the sterol structure. Now, that carbon 25 has normally one hydrogen coming off it. It's got 3 bonds to other carbon atoms, but then it will have one final bond to a hydrogen atom. We're going to remove that bond to the hydrogen atom and replace it with a bond to an alcohol group instead, and therefore we will have added a hydroxyl group onto the 25th carbon, and that is what is meant by 25-hydroxylation of vitamin D. Okay, so we're going to uptake the vitamin D, which I'll now just abbreviate even shorter to D here, and we're going to convert it into 25-hydroxy vitamin D, which for short is written like so, 25 dash and then hydroxy, OH for hydroxy, and then D like so. So that's the shorthand for 25-hydroxy vitamin D. But I suppose I should write it out in full. So in full, 25-hydroxy vitamin D is what this stands for. And of course, again, you can have 25-hydroxy vitamin D2 and 25-hydroxy vitamin D3. This covers both of them. So 25-hydroxy vitamin D is this umbrella term for both 25-hydroxy vitamin D2 and 25-hydroxy vitamin D3. Okay, so both of them are going to be converted into the 25-hydroxy form, and then of course this will be released back into the blood, and again it will be carried around the blood by the vitamin D binding proteins. Okay, so vitamin D is converted then into 25-hydroxy vitamin D, and this process is known as 25-hydroxylation. Now that makes it a little bit more active, but it's still not really in the active state. Okay, 25-hydroxy vitamin D is still not particularly active, but it's closer to being in the active state. The final process that needs to occur in order to turn the vitamin D from being in an inactive form to being in an active form is the process known as 1-hydroxylation. And 1-hydroxylation is a process that is not normally turned on. So normally the 25-hydroxy vitamin D molecules in the blood are stuck. They cannot go any further in their activation process because 1-hydroxylation is not available to them. Now, 1-hydroxylation is performed in the cells of the proximal convoluted tubule. Okay, so this cell here, where 25-hydroxylation was occurring, was a cell of the liver, a hepatocyte, whereas 1-hydroxylation is going to occur in a renal tubule cell in the proximal convoluted tubule. So let me just draw one of these renal tubule cells here. So here is the renal tubule cell, like so. And I'll just label it up. So this is a proximal convoluted tubule cell here. Okay, now it is not usually actually going to have the 1-hydroxylation process turned on. It is the cell in the body which can actually take 25-hydroxy vitamin D molecules and convert them into the active form of vitamin D, but usually it doesn't bother doing it. However, when parathyroid hormone comes down and stimulates the parathyroid hormone 1 receptor, this G protein coupled receptor on the basolateral membrane of the proximal convoluted tubule cells, what's going to happen is they're going to turn on the 1-hydroxylation process. So here is the parathyroid hormone 1 receptor, here is parathyroid hormone coming in and stimulating it. Now 1-hydroxylation is going to be turned on, and the 25-hydroxy vitamin D molecules that the proximal convoluted tubule cell is exposed to are now going to be converted into 125-dihydroxy vitamin D molecules, okay, which is short written like so, 125 dihydroxy, so we put alcohol 2, and then D for dihydroxyvitamin D here. And those molecules are then going to be released back into the bloodstream, okay, and now you're going to have the final active form of vitamin D appearing in the bloodstream, the 1,25-dihydroxyvitamin D, and this is what's actually going to do something. So when parathyroid hormone is released by the parathyroid glands in response to hypocalcaemia, it's going to activate the cells of the proximal convoluted tubule to begin the process of 1-hydroxylation to activate 25-hydroxyvitamin D molecules in the bloodstream, which are inactive to all extents and purposes, into the actual active form of vitamin D, which is this 1,25-dihydroxyvitamin D, written like so for short. Okay, so writing out the full name then, this is 1,25-dihydroxy, and then it should be all one word, but I've run out of space, so I'll put it underneath here, vitamin D. So that's the full name for this molecule here. And again, you can do this for vitamin D2 or vitamin D3, so you can have 1,25-dihydroxyvitamin D2 or 1,25-dihydroxyvitamin D3. Okay, so going back to the structures, what does it actually mean? Well, carbon number 1 is this carbon in the case of vitamin D3, or this carbon in the case of vitamin D2. So we can see that it has 2 hydrogens coming off it, normally. What we're now going to do is take one of those hydrogens off and replace it with an alcohol group to make the 1-hydroxy form here. Okay, and now we have vitamin D molecules with 2 hydroxy groups added onto them, one off carbon 1 and one off carbon 25, so these are now called 1,25-dihydroxyvitamin D molecules, and these are the active forms, whether we're talking about vitamin D2 or vitamin D3. So again, I'll stress it, D2 and D3 are utterly equivalent as far as the body's concerned, which is why we're just umbrella terming them as vitamin D. Okay, so parathyroid hormone then triggers the production of this active form of vitamin D, 125, that was an awful mistake, dihydroxyvitamin D. So we're now going to get 125-dihydroxyvitamin D appearing in the bloodstream in response to the parathyroid hormone. And this 125-dihydroxyvitamin D is going to work with the parathyroid hormone to try and bring calcium levels in the body back up. Specifically, free calcium levels is what we're trying to get back up. Okay, so what I'm now going to do is I'm going to start with one of the first effects that I'd like to show you of what 125-dihydroxyvitamin D is actually going to do, because it's actually going to have a negative feedback effect. It's actually going to go back up to the parathyroid glands, and it's going to stop further synthesis of parathyroid hormone. Okay, this is going to be a negative feedback loop. So we don't want the parathyroid glands to get carried away. We don't want them to release too much parathyroid hormone, otherwise we might end up with blood free calcium level getting too high. Okay, and too high free calcium ion concentration would be detrimental to intracellular signaling as well. So once we've actually produced the 125-dihydroxyvitamin D, and therefore parathyroid hormone is evidently starting to have an effect on the body, we need to tell the parathyroid glands to stop releasing it now. We've got the parathyroid hormone, we're now beginning to actually see the actions of this, let's stop making any more. And indeed, 125-dihydroxyvitamin D is going to be part of that negative feedback loop. It's going to go up to the parathyroid glands and tell them now, shut down your synthesis of parathyroid hormone. We've got enough in the blood now. We've got enough of this effect occurring. Okay, and this will be a good way of actually showing you the receptor for 125-dihydroxyvitamin D and how therefore this active form of vitamin D is going to affect cells. So we're going to kill two birds with one stone. This is a nice introductory example as to how 125-dihydroxyvitamin D is actually going to act, and it's also part of the story, the negative feedback loop. Okay, so I'll just put the title, Negative Feedback, and then we'll actually draw a picture for this. So there is a receptor for 125-dihydroxyvitamin D, and it is a nuclear receptor. Okay, so it's going to be a receptor that when the ligand for this receptor binds, is going to actually affect the transcription of genes within the cell. It's going to affect gene expression within the cell. So, negative feedback. Right, so where to begin? So I think the first thing to say is that 1,25-dihydroxyvitamin D, it's still reasonably lipid soluble, so it can still diffuse through cell membranes and through nuclear membranes. So it's going to, now that it has risen in the blood, end up inside the chief cells of the parathyroid glands. So it's going to go back up to the parathyroid glands, it's going to go into those cells, and it's going to end up in the nuclei of those chief cells of the parathyroid gland. So what I'm going to now do is I'm going to draw a nice big picture for the nucleus of one of the chief cells of the parathyroid gland here. Okay, so let me just make sure this is all nice and straight here. So this is a nucleus of a chief cell of the parathyroid gland. Okay, so I'll just label it up as nucleus. Now what's going to happen is our 1,25-dihydroxyvitamin D is going to end up in the nucleus of the chief cell here. So we'll have 1,25-dihydroxyvitamin D coming in, and I'll just have it represented by a little blob here. So this little square, or little rectangle here, is going to represent the 1,25-dihydroxyvitamin D. So it's come into the cell, into the chief cell, it's now gone into the nucleus of the chief cell, and in the nucleus it's going to bind to its receptor, which is found in the nucleus and is a nuclear receptor. So now it's going to bind to its receptor here, and its receptor is called the vitamin D receptor. So this rectangle here, this is called the vitamin D receptor, and for short we'll abbreviate that down to VDR. So VDR stands for vitamin D receptor. So we'll have the vitamin D receptor coloured in blue, like so. So 1,25-dihydroxyvitamin D is going to bind to the vitamin D receptor, and now the vitamin D receptor is going to heterodimerize with another really important nuclear receptor known as the retinoid X receptor. Okay, so I'll show this here. So here, this is the retinoid X receptor, which for short is often abbreviated down to RXR. So I'll write out in full what this stands for. So this is the retinoid X receptor. And for those of you who have seen pathways involving nuclear receptors before, you might well have seen the retinoid X receptor before. It plays a big role in nuclear receptor pathways. Okay, so the retinoid X receptor, which we'll have in orange here. Okay, so once the 1,25-dihydroxyvitamin D then has bound to the vitamin D receptor, that will activate the vitamin D receptor, and it's now capable of binding to this retinoid X receptor. So they're going to form a heterodimer. So these two are going to come together, and I'll just show the heterodimer here. So here is our heterodimer, like so, and I'll just colour code them in. So here we have the retinoid X receptor, here we have our vitamin D receptor in blue, and of course bound to the vitamin D receptor we're still going to have our activated form of vitamin D, the 1,25-dihydroxyvitamin D, and I'll just give it a boundary. Okay, right, so now what is this heterodimer going to actually do? Well, of course, it's going to go and bind to a certain sequence of DNA. So there'll be a certain sequence of organic bases, and I don't actually know what this specific sequence of organic bases is for this specific heterodimer here, but there will be a specific sequence of organic bases, usually around 8 to 10 in length, that this complex here will now be capable of binding, and that sequence is known as the cis-regulatory sequence for this transcription factor. Okay, and what will now happen is you will find this cis-regulatory sequence in the gene control region of a huge number of different genes in the genome, and those are the genes that are going to actually have their expression level regulated by the vitamin D receptor. Okay, now, one of the genes which has the cis-regulatory sequence for this complex of the vitamin D receptor with the retinoid X receptor is actually the parathyroid hormone gene. So let me now draw this here. So here, let's say this is the gene for the parathyroid hormone. Okay, so I'll have it in pink here. So this is the parathyroid hormone gene. Okay, and now, in the gene control region upstream of the parathyroid hormone gene, we're going to have a cis-regulatory sequence for this complex here. So now let me just add on, and I'll do it in yellow. I think yellow is the best colour here. this is the portion of DNA upstream of the parathyroid hormone gene, and of course this is the region that controls the expression, controls the transcription level of the downstream gene, and it's known as the gene control region. Now, the portion of the gene control region immediately upstream of the parathyroid hormone gene, this portion here if you like, that will be what is known as the actual promoter region. And of course the promoter region is where the general transcription factors bind and assemble into a complex, and then the enzyme RNA polymerase actually binds on top of that and then will make its way along the gene and transcribe the gene into mRNA. So this is known as the promoter region. So that's the portion of the gene control region immediately upstream of the parathyroid hormone gene. But then the gene control region goes on for a much longer portion past the promoter region here. And in this portion, you're going to have cis-regulatory sequences for all sorts of different other transcription factors known as specific transcription factors. So remember, there are two different types of transcription factors: specific transcription factors and general transcription factors. General transcription factors are those ones that bind to the promoter regions upstream of every single gene in the genome, okay? And they have to bind there and assemble into a complex in order for the RNA polymerase enzyme to bind on top. So examples of things like transcription factor 2D. So those are what are known as the general transcription factors, and I'll just abbreviate them down to the general TFs. This complex of retinoid X receptor with the vitamin D receptor, this is an example of a specific transcription factor. And these bind to little sequences of DNA known as cis-regulatory sequences, which can be found in the gene control regions upstream of the promoter region, but the portion of the gene control region upstream of the promoter region in certain, upstream of certain genes. Okay, so for certain genes. So for this specific transcription factor, there will be a whole bunch of genes that have a cis-regulatory sequence for this specific transcription factor upstream of them, i.e. in their gene control region. The parathyroid hormone gene is one of them, so it will now have a region for this complex to bind. So this complex will bind here, let's say, so this is the cis-regulatory sequence for this complex of the vitamin D receptor with the retinoid X receptor. And now what it's going to do is it's going to affect the likelihood that the downstream gene is transcribed. And in the case of the parathyroid hormone gene, it's actually going to decrease the probability that this is going to be transcribed. Now, how can it do that? Well, firstly, it can directly bind to the general transcription factors as they attempt to assemble on the promoter region and make it less likely that they're actually going to assemble completely there, and therefore less likely that the RNA polymerase enzyme will bind on top of them. Or alternatively, they can affect the chromatin structure. They can affect how tightly the DNA is actually wrapped around the histone complexes. Okay, they can make the DNA wrap around the histone complexes tighter, and when DNA is wrapped around histone complexes tighter, it's less likely that you're actually going to get the general transcription factors assembling on the promoter region here with the RNA polymerase enzyme assembling on top, i.e. transcription becomes less likely. So overall then, to summarise, when the activated form of vitamin D, 1,25-dihydroxyvitamin D, binds to the vitamin D receptor, that vitamin D receptor is going to change conformation so that it can complex with the retinoid X receptor, which is also in the nucleus, and this heterodimer of the vitamin D receptor with the retinoid X receptor is then going to bind to a specific sequence of DNA known as the cis-regulatory sequence for that specific transcription factor, and this cis-regulatory sequence for this complex here is in fact found in the gene control region upstream of the parathyroid hormone gene, and thereby the vitamin D receptor with the retinoid X receptor can actually affect transcription of the parathyroid hormone gene, and it's going to repress it. Okay, so transcription of the parathyroid hormone gene is going to go down, and therefore you're going to produce less parathyroid hormone. So vitamin D overall is going to inhibit the synthesis of parathyroid hormone in the chief cells of the parathyroid gland. Right. So that's specifically how it's going to have this negative feedback role, i.e. turning off synthesis of parathyroid hormone in the chief cells of the parathyroid gland. However, this is more general. This is the receptor for the activated form of vitamin D. So whenever we talk about the effects of vitamin D, and we're going to see other effects of vitamin D, from now on, this is how it acts. It acts on the vitamin D receptor, which is a nuclear receptor inside cells. It will complex once it's bound to the vitamin D with retinoid X receptor, and that vitamin D receptor retinoid X receptor heterodimer is then going to act as a specific transcription factor. And what do those do? They change gene expression within cells, and that is how they change the behavior within cells. So vitamin D works to change behavior within cells by activating a specific transcription factor, which is then going to change gene expression within those cells. And here is an example of it changing the behavior of the chief cells of the parathyroid glands and preventing them from synthesizing further parathyroid hormone. Okay, so we'll have a break here, and in the next video what we'll talk about is the combined effect of parathyroid hormone and vitamin D on bone to try and release calcium from the bone.